Hello students, this is Dr. Faizan Mirza. In this video, I'll be covering with you the concept of phagocytosis that already I've covered in one of my videos. I'll be recalling it and we'll be discussing this question from October-November variant 23 from 2022. So what do we know about lysosomes? What do we know about phagocytosis? So if you recall, we know the lysosomes, their general diameter is about zero point 0.1 to zero point 0.5 micrometer, and they are spherical sacs. They have a membrane around them, and they arise from Golgi vesicles only. And they have up to 50 different hydrolytic enzymes, and these hydrolytic enzymes these include carbohydrases, lipases, these include DNases, RNases. You can just name the biological molecule and add A's to it because these lysosomes will have these enzymes that can digest these biological molecules. All of them are there inside the lysosomes, inside this vesicle, and they can digest all the type of biological molecules presented to them, including the harmful chemicals as well. So this is for example the membrane of a white blood cell, a phagocyte, how it how it carries out the process of phagocytosis. So we know that there are, for example, bacteria outside this phagocyte, and inside the phagocyte there's a lysosome, and the lysosome will have hydrolytic enzymes, the ones that I just named, and a lot more, and they are all there. They all are separated from the environment of the cytoplasm by the help of this membrane. Now what will happen, the bacteria starts to get phagocytized into the cell. And how phagocytosis occur by this bacteria binding to the receptors present on the cell surface membrane of the phagocyte. The membrane starts to form this kind of a structure in which it starts to surround the bacteria. The membrane encircles it. The membrane encloses this bacteria and pulls it inside into the cytoplasm. Once it's inside, it is bound within the vesicle. We call this vesicle phagosome. If asked for the name, you can state. Otherwise, mentioning the name in a question of description or explanation, the term phagosome will not get you a mark, because if they have not asked for this term, then this term will not fetch you any marks. So anyhow, the phagosome starts to move within the cytoplasm, and the vesicle, the lysosome, starts to move as well. They both fuse together. They both fuse together to form the structure called phagolysosome. The lysosome then start to the lysosomal enzyme starts to digest this bacteria. The digested bacteria is this it's again surrounded in the same membrane of the lysosome, and this structure, and the contents which are here, they are assimilated into the cytoplasm, the ones that the cell can use, and the debris which the cell cannot use will start to move towards the cell surface membrane again, and this is all the debris that is present inside it, and the vesicle will go and fuse with the cell surface membrane, and exocytosis of the debris occur. When exocytosis occur, this debris is released out. These are some, for example, some 7 stages or 8 stages, in fact, of how the engulfing occur, how it moved into the cytoplasm, how it fused, how the lysosomal enzyme started to act, how this bacteria got digested and the product got assimilated, and then the vesicles started to move towards the carry the debris towards the cell surface membrane and fuse it again, and exocytosis occur. The pathway that these vesicles actually are following are all the microtubules, because microtubules are the passageways, or in fact the pathways, not the passageways, the pathways on which the structures within the cell can actually attach and then travel along in the cytoplasm, and this uses ATP. So keeping all this information in mind, just recalling it, let's come to the question that came in October November 22 in the variant 23. And the question is that they give you a scanning electron micrograph showing a macrophage engulfing some bacteria. So these are the bacteria, these rod-like structures, these are the bacteria, and this is the phagocyte there, and this is the phagocyte forming an encircled structure so that the bacteria can be pulled inside into the cytoplasm of the phagocyte. Describe how macrophage engulfs bacteria. Now you have to give description, so you need to understand that stating a term will not get you marks. So what points can you add for 3 marks? There are antigens on the cell surface membrane of bacteria. Antibodies bind to these. Macrophages carry out phagocytosis of these bacteria. The membrane wraps around the bacteria. You will include the term wrap because this will fetch you marks. and infolds into the cytoplasm. Instead of using infold, you can use the term of invaginate as well. It invaginates or infolds or pinches in. You can also use the term of pinches in into the cytoplasm. The membrane of the macrophage fuses to form a vesicle. The fusion of this vesicle, the membrane, to form a vesicle will fetch you a mark as well. And then the phagosome is formed. I have mentioned the phagosome in parentheses because this will not get you a mark, although I mentioned this to just make sure that you connect with the term with the bacteria enclosed, and this uses ATP. So this is the description provided. This is the description for this question. Explain the function of lysosomes in cells such as macrophages. Lysosomes are membrane-bound organelles that have hydrolytic enzymes such as proteases, nucleases, hydrocarbohydrases present inside. The membrane of the lysosome keeps these enzymes separated from the cytoplasm. The function of the lysosome is to fuse with the phagocytic vesicle, having the bacteria or the old worn out organelles present inside the macrophage. This will give you enough marks. Moving on, the question then moves on to tuberculosis. Tuberculosis cases are provided to you in USA from 1993 to 2018. This is 1993, this is 2018. Number of new cases per 100,000 of population and the number of new cases as well. So this is the information provided to you for TB. So before going on, let's just recall what we know about TB. So we can recall what the TB was. TB is caused by bacteria, rod-shaped bacteria, and the rod-shaped bacteria could be Mycobacterium tuberculosis, the causing TB in the human host, Mycobacterium bovis causing in the cattle host, and this disease spread by droplet transmission. If the people are living in close proximity, overcrowded population, or there's an increased chance of passing the bacteria from one person to another. If the person is if an individual is using undercooked meat or unpasteurized milk, then the M. bovis, which is causing the big TB in cattle, can pass on to the humans as well. And the disease takes a long time to develop. And these are the basic signs and symptoms of the disease. The treatment include isoniazid and rifampicin at an antibiotic course that you have to carry out for about 6 to 9 months. Two antibiotics have to be taken, and it's a prolonged course. And vaccination is available for BCG. Worldwide distribution, there are a lot of anti-TB drugs available, but the TB drug resistant strains are also there, and MDR-TB, XDR-TB. And primary infection is in lungs, but the disease can spread to other systems through blood. It can go to the digestive system, the reproductive system, and all. So yeah, that can occur. And in the infectious disease, we have also seen that when we are discussing the statistics of any incidence or the statistics of the diseases, how the diseases are studied. Incidence means the number of new cases diagnosed over a certain period of time. Prevalence, the number of people who are currently suffering from the disease. Mortality rate is the number of deaths over a particular period of time. Data. Normally, when we collect the data for any infectious disease, the data is collected and represented as per 100,000 individuals. Why per 100,000 individuals? Because it's easier for follow-up and comparison between different times and different parts of the world. So accordingly, the medication for example, vaccines or the hospital facilities and the beds can be made available. Why I'm discussing this? Because this is exactly where the question leads, the question that I was just discussing. I'm still on the same question, the next part. So this is the number of new cases of TB shown to you. In 1993, they have shown you the cases till 2018. You can see the overall trend that the cases decreased, and then after 2001 there is a slight decrease, and the decrease is then gradual, and then it becomes plateaued. For example, you can see in this case from 2013 to 2018 there is a plateau. There is a certain rise in 2015. This rise probably could be because of drug resistant strains being developed, and you can see number of new cases are actually shown to you by as per 100,000 of population, as already mentioned and shared, that why is it useful. Calculate the percentage change in the number of new cases on TB between 1993 and 2018. You will apply the formula percentage change final minus initial upon initial multiplied by 100%. In this case, initial is the value of 1993 and the value of final is the value of 2018. So you read off the values of 1993 from here and the one from 2018 here, and these value are 9,000 for the final value, the 2018, and 25,000 for the initial value. You have to make sure that you read off the values particularly very in very precision. With precision, you should draw a line with the scale, so you read off the actual point there. You know, if this is the point they are asking, you can divide it into equal four parts. So if this is 5,000, this is 10,000, so these must be 4 divisions, and they are asking this division, so this must be 9,000. So you just subtract 9,000 from 25,000, divide by the initial value 25,000, you get the answer of minus 64%. If the answer comes negative, yes, you will include the sign of negative, although the negative sign will not get you an extra mark, but yes, since it's a percentage decrease, so minus sign can be added since you are subtracting this 9,000 and minus 25,000 from here. So larger value is being subtracted from the smaller value. Anyhow, so the answer comes out to be 64%. You will get 3 marks from here. Use the figure 2.2 to describe the trend and number of new cases between this and this. Is what actually you are seeing there. So the data show the number of new cases showed an overall decrease. Overall trend you should just state a much steep decrease is seen between 1993 and 2001. Then the number of new cases between 2013 and 18 is a bit constant. There's a plateau in 2015. New cases increased slightly. So this is something that you can state. So just an advantage of calculating number of 100 cases per 100,000 already discussed with you for a valid comparison between different times and different places. High incidence areas, countries, for example, TB can be highlighted accordingly. Drugs, vaccines can be made available. TB is endemic, always present in many populations in the country. State tourism why is it difficult to reduce the number of TB because there are people living in overcrowded conditions. There is a high chance of disease being transmitted from one person to another using from the airborne droplets or from using unpasteurized milk. And the transmission from cattle to horse is common as well. Then you can also include the point for antibiotic being more than one antibiotic is to be used and the treatment lasts a long time. So that actually leads to people not completing the course and the antibiotic resistant strains develop and transmission increase as well. A similar question then came in Feb-March 2023, variant 22, in which they asked for tuberculosis. Tuberculosis and influenza, polio are example of infectious diseases. They generally just ask you explain the term what is infectious disease. So a disease that is caused by a pathogen is called an infectious disease. Infectious diseases can be transmitted from one individual to another, either through direct contact or indirect contact with the infected individual. Name the species organism that cause TB. So Mycobacterium tuberculosis can be stated. Mycobacterium bovis can be added because they're asking a species. They have not specified whether cattle or host. The host is cattle or the host is human. Immunity can be described as artificial active or natural passive. Natural and passive or active. Name the type of immunity that mother gives to her baby through breast milk. That's natural. Passive, something of a fact. The influenza virus can mutate frequently to produce different strains of the virus. A new vaccine is often necessary to stimulate the production of new antibodies to the new strain. Explain why new antibodies are to be produced and to give immunity to the new strain. We know that vaccines they provide you with memory cells in which the antibodies receptors are there which have a variable region complementary to which the antigen can bind. So when vaccines are given, these antibodies are produced, the memory cells are there, they can convert to plasma cells, and when the antigen enters they can identify it without clonal selection and clonal expansion, and the disease can be cleared off. So why different antibodies need to be produced to give immunity against the new strains? We know this, the new vaccines will have the antigens of the newer strains because the strains will be different. Different strains of the same virus have different antigens. Antibodies that recognize the previous variant will not recognize the new antigen. The new antibodies will have to be produced that have the variable region complementary to the new strains of the antigen. You need to recall how the antigen antibody complementary they bind to one another. An antibody recognizing one antigen will not bind to another antigen. You need to have a separate antibody for that. Then they move on to polio. It's a serious viral infection. In 1986, polio caused paralysis more than 75,000 children across Africa. Explain how vaccination programs can help to control spread of infectious diseases such as polio. We know that vaccination programs help in breaking the transmission cycle. The most important thing, they break the transmission cycle. Multiple doses of vaccine help the immune system to have the memory B cell having receptors to recognize the pathogen of polio or another infectious disease whichever being vaccinated for. Vaccinating large number of people provide herd immunity. In herd immunity, when large number of people have been vaccinated, the unvaccinated people are protected because the chance of disease transmission is lowered. So these points can be added for 3 marks. Antibiotics such as penicillin do not help prevent the spread of viral disease such as polio. Explain why penicillin is not effective against viruses. Penicillin, you need to recall where it acts on the cell wall. It acts on the growing cell because when the autolysins were breaking the crosslinks of the peptidoglycan cell wall, the crosslinks are not remade if the penicillin is being used. You can just recall the fact that you have studied. So why the virus is not here using penicillin? Because virus lack the target site cell wall For the penicillin, it does not have peptidoglycan. You can include the term peptidoglycan here. There is no peptidoglycan cell wall because they are asking you the why is it not effective. Plus, you can also state this drug is affecting growing cells only. Although 1 mark answer, it needs 1 point, but both points can be added. Obviously, they will just they tell you the first mark point, so your first point must be the most relevant one. I hope this video was helpful for you. That's it from my side. Thank you so much.